Continuing on with the cerebrum. The association areas of those, we've talked about different features and different things of the lobes, how they do different tasks, and they overlap in some cases. So, for example, the frontal lobes, they have some big important things to do. They are responsible for our higher intellectual processes, the things we think of as thinking. So things like concentration, planning, problem solving, or even potentially what we call the moral compass or judgment of behavior. Psychologists debate about it all the time, whether or not it actually exists. We've never found an actual portion of the frontal lobes that does that task. It's been debated for centuries, and people still haven't come to a conclusive answer on that. But when we talk about other animals like a mouse or a bird or something like that, sometimes people say, well, they don't have all these tasks. Sometimes they do, sometimes they don't, but generally they don't. A fish probably doesn't have most of these tasks because they don't have this frontal region in large space of their cerebrum, therefore they don't have that ability, whereas we do. So we have all these kind of things. And people have sought to adjust that behavior through things like doing chemicals or drugs in order to affect the way this works, because maybe it's not working correctly, or even back into earlier parts of the 1900s where people did things like lobotomy, where they went in and they basically tried to damage the frontal lobes in order to make them function with less problems, basically. We've noted today that that's not a very accurate way to do it, just randomly sort of destroying the frontal lobes. That's not really an effective medical procedure. The parietal lobes, understanding speech and choosing our words, our verbal skills. Our temporal lobes, our memory of how visual scenes, music, and patterns work. Sometimes you may have seen a picture and said, you know what, I saw this picture before of now when I see it, every time I see that picture, I think of a particular song because I heard the song at the same time I saw it the first time. That's because a lot of times these things are stored very similar to each other. And so our brain connects them as a pattern. However, occipital lobes are responsible for visual pattern recognition. So I look at an object and go, oh yeah, that's an apple. I know that because I know that pattern in my brain. Now, could I fool it by taking another type of object similar to it and painting it the same color as an apple, and then my brain thinks it's an apple and it's actually a different fruit? Absolutely. But our brain will actually first think it's that particular thing because that's what our brain's pattern recognition does. It goes right to that first thing. The general interpretive area is located where the parietal, temporal, and occipital lobes meet because they have to connect all information to generate a general view of the world of how things are going. So for example, this general interpretive area is right there. The hemisphere domination. Before I talked about the fact that people talk about people being right brain or left brain. And really, you're both brained. But one can be more dominant than the other for certain tasks. So for example, left is dominant for about 90% of people for speech, writing, and reading. So your verbal skills, your language skills, are generally dominated by one side of the brain, and that's usually the left for most people, and the other 10% would be right dominant. Does that mean it's connected to your right-handed versus left-handed? Always the question people ask. The question to that is no. Even though it's about 90% right-handed people, 10% left-handed people in the world, they're not actually connected. You can have people who are left-handed and left-dominant, or right-handed and right-dominant, which actually should be the opposite way if it was the way the case worked. Theoretically, if you were left-dominant, you would be right-handed because that side of the brain controls that hand. Handedness is thought to be something resulting from an internal thing within the womb that causes us to choose one or the other, and we typically choose right. Non-dominant hemisphere is still doing things. It's not just sitting there wasting time or doing nothing. It has tasks. First off, nonverbal functions. It does a lot of things like how's our body oriented in space? How's it dealing with things? How's somebody, when we look at things, how are those set up? Understanding musical patterns work really well with this. Nonverbal experiences. So I see a hand gesture. I see a particular thing or how a person's body posture is, or how their facial expressions are. A lot of that's being done by the non-dominant side, not the dominant side, so it can work together for us to get a picture of what are they saying plus what are they doing with their body, working together as one picture. The dominant hemisphere controls both sides for movement, for motor functions, because it makes sense. If one side is doing this, the other side is trying to do something else at a different time, they're not going to be coordinated very well. So I need one side to make the decisions and coordinate both sides. So for most people, it's going to be the left side is going to tell the whole body what to do. Even though the right side is still controlling the left side, right side is controlling the left side is controlling the right side, this side is controlling both. It's telling both sides what to do at the same time. You also have basal ganglia or basal nuclei. These are groups of gray matter inside the white matter towards the base of the cerebrum. Their job is to relay information. So they're actually a relay station. If there's problems with them, that can disrupt the signals. So for example, they're located right here. If something's coming through here and that gets disrupted, I could either send too many signals or not send enough signals and cause problems both ways. How does that work? Well, typically dopamine works, one of the neurotransmitters. It actually has a role in helping to control signals coming through the basal ganglia. If there's problems with dopamine, it can affect too much signal or too little signal by how it inhibits those signals. So some examples of diseases are famous for causing problems with basal ganglia. Parkinson's. Parkinson's, one of its key features people know is things like shakes and tremors that are uncontrollable. The reason why is really simple. There are very low dopamine levels. Dopamine is going to inhibit the signals, causing them to be controlled. In this case, when there's not the control there, there's too many signals going through, and basically they just keep shaking because their brain's sending constant signals, even though there's really not a need to do it. Normally that would be controlled by the dopamine levels, but since there are no dopamine there, or low levels, it allows all the signals to go through and they constantly send little tremors. Another one is Huntington's disease, very horrible disease, which is a genetic disorder. It's dominant one. If you have any copy of it, you have the disease, which progressively causes you to lose the ability to use your muscles until eventually your organs shut down, your muscles are all gone, you can't move, you're bedridden, and you die from organ failure. The concept here is the basal ganglia gradually deteriorate. As they deteriorate, you lose progressive ability to use your muscles because the signals aren't getting through anymore. And eventually it's going to shut down all use of the muscles. The muscles will atrophy and basically will wither away, and you will be bedridden and can't do anything until eventually organ failure. Inside of the cerebrum are ventricles. Ventricles are these little chambers that hold the cerebrospinal fluid. So what does the cerebrospinal fluid actually do? It nourishes the brain and removes waste. It acts like the blood. I don't want blood inside the nervous tissue because it carries too many things like pathogens that can cause problems. So what you do is, I want the nutrients from the blood. So I'm going to take the plasma, I'm going to convert it to cerebrospinal fluid, which is what happens within this system. I take it and I allow it to be transformed and changed into cerebrospinal fluid, which can then flow through the system and protect the system. It's also going to cushion some nervous systems. So think about subarachnoid space full of fluid. As my brain moves, it acts like a little bit of a shock absorber to keep it from being damaged. The ventricles themselves, there are 4 of them. Theoretically, we'd number all 4 of them, which we do, but the first 2 are kind of parallel to each other. So these are cavities. They're continuous with the subarachnoid space. They're continuous with the central canal, allowing the fluid to flow through the whole area. So the first 2 are called the lateral ventricles. You can think of them as number 1, number 2, for whichever side of the brain you want to talk to them about, but they are equal to each other because they're both side by side at the base of the cerebrum. The next one is the third ventricle. Well, if you can guess what the last one's called, it's not third ventricle, it's going to be the fourth ventricle. I have to connect these up. I have to have a way to have something go between them to allow the fluid to pass. So between the lateral and the third ventricle is what's called the foramen and Monro. A foramen and Monro is a Y-shaped connection that allows them to come down like this, 2 lateral, connected together, come down to the third, allow stuff to come through. That means I also have something go to the third or the fourth. That would be the cerebral aqueduct. So it's going to go between the third and fourth ventricles, connecting up and allowing fluid to transfer through that area. You can see it here from a view where the brain's been tilted back to look at it this way. These horn shapes are the lateral ventricles, the first 2. Here's your foramen and Monro, or interventricular foramen, and it's also here the third ventricle, the cerebral aqueduct, and then the fourth ventricle, and then down into the spinal cord. Side view shows the same basic principle here. The choroid plexus is a region of capillaries, a network of them. They're carrying blood, they're the smallest blood vessels. What's going to happen is you're going to allow blood to filter the plasma out of there, and it's going to go through this region and become cerebrospinal fluid and empty into the lateral ventricles. So these are located in the walls of the lateral ventricles. This forms what we call a blood-brain barrier, allowing what's in the blood, whether it be positive or negative things, toxins, things like that, for the most part, they're going to stay in the blood and not come across this barrier. Does that mean everything can't get through? No. Things like alcohol get through this barrier, they're able to pass through it. Are there types of viruses that can make it through? Absolutely. Are there bacteria that can make it through? Some can, most of them cannot. So it usually is protective, but sometimes it is not able to do its job and causes problems. And unfortunately, we don't have all the other things there, like white blood cells, to help protect the nervous tissue, so they do become more vulnerable because they don't have it in there because you're not allowing them to pass across this blood-brain barrier. The cerebrospinal fluid flows in one direction. It's going to follow a pathway through the whole system. That way, it doesn't go back upon itself and bring waste products back to the area that it just cleared out. Starts at the choroid plexus. It's going to generate the cerebrospinal fluid. It's going to go to the lateral ventricles, where it's going to be dumped into. From there, it's going to go to the third ventricle through the foramen Monroe. It's going to go from there to the fourth ventricle, the cerebral aqueduct. Here's the proverbial fork in the road. Some of it's going to go straight into the central canal, right down the center of the spinal cord. Some of it's going to go over into the subarachnoid space and start to flow around that way. And eventually, the central canal is going to connect back up to the subarachnoid space. So I come here, I fork out, and this is going to come back and reconnect. At the end, both these have to leave this system and go back to where they came from, which in this case is the bloodstream. I want to return there. That way all that waste product can be taken through the system and cleaned out just like you would clean out your normal blood supply. So it's going to follow the same basic principles that way. So you see it's flowing through here, coming down this way. Here, and it's going to hit that little spot where it goes this way in the subarachnoid space or straight down the central canal. And that